Okay, let's unpack this. You've brought us some really fascinating sources today. We're diving into the absolute cutting edge of oncology research with a big focus on prostate cancer.
That's right. And it's a great mix, I think. We've got things happening right now in the clinic, immediate applications, but also some revolutionary stuff still in the lab phase.
Exactly. So our mission here is to kind of connect those dots for you, the listener. Look at how doctors are using aggressive treatments now, while scientists are simultaneously using tools like, well, AI and metabolic reengineering to basically reshape future treatments.
Yeah, it's quite the spectrum. We'll start with the clinical trials, what's working for patients today, and then pivot to let's say, the next generation of discoveries in the preclinical space.
Sounds like a plan. So kicking off with the clinical side, aggressive multimodal therapy. Tell us about that study.
Right, so this was an interim look at a clinical study. They were focused on men with what's called polymetastatic prostate cancer. Basically, 4 or more bone metastases, so pretty advanced disease.
Okay, high volume disease, and multimodal means they threw a lot at it.
They did. It was a combination of 4 treatments up front. They used enzalutamide, that's an RRSI, an androgen receptor signaling inhibitor, plus denosumab, and then also local radiotherapy, or LRT, and metastasis-directed therapy, MDT.
Wow, okay. That is aggressive. So how did it pan out for the 20 patients in this interim analysis?
The results were pretty encouraging, actually. 1-year progression-free survival PFS was 84.4%.
That's quite high for this stage.
It is. And maybe even more importantly, half the patients achieve a PSA level below 0.2 nanograms per milliliter. That's considered a really strong surrogate marker for better overall survival down the line.
So it definitely supports that idea of hitting the cancer hard and early, even when it's already spread significantly.
Precisely. It backs up that comprehensive upfront strategy.
Okay, so that's combination therapy, but then there's the balance trial. This one really leans into personalization, using something called PAM50.
Yeah, the balance trial is fascinating. PAM50 is a gene expression tool, essentially. They used it to subtype the tumors, to classify them based on their genetic activity.
And why was that subtyping so critical?
Because it predicted who actually benefited from adding a drug called Apalutamide to their salvage radiation therapy. It wasn't for everyone.
Ah, okay. So what did they find? Which group saw the benefit?
The key finding was in patients with a luminal B tumor subtype. That was about 43% of the men in the trial. For them, adding apalutamide made a huge difference.
Okay, here's where it gets really interesting. As you said, what was that difference quantitatively?
For the luminal B group, the 5-year metastasis-free survival was 94.7% with apalutamide.
94.7.
Wow.
Yeah.
Compared to 81.8% for those who got the placebo with their radiation.
That's a major difference. And what about the non-luminal B patients?
That's the other crucial part. The non-luminal B patients didn't get a clear benefit from adding apalutamide. So, if you didn't do the PAM50 subtyping first.
You'd be giving a potentially toxic and expensive drug to over half the patients for no real gain.
Exactly. It really proves the power of using genomics to guide treatment selection. True personalized medicine in action.
Absolutely. Okay, so those trials show what's possible now. But let's shift gears, like you said, and go preclinical into the engine room of the cancer cell itself.
Right. We're moving into metabolic reengineering, specifically targeting a mitochondrial protein called ANT2. That's ANT2, adenine nucleotide translocase 2.
ANT2. And why target that?
Because it's highly overexpressed in prostate cancer tissue, sometimes up to 4 times the normal level. Think of it like a specific fuel transporter that the cancer cells are really dependent on.
Okay, so it's helping fuel the cancer. What happens if you block it?
Well, this is where it gets cool. When researchers blocked ANT2, it dramatically amplified the activity of T cells, the immune system's cancer fighters.
How did it do that?
It seems to rewire their energy metabolism. Blocking ANT2 boosted the T cells' ability to proliferate and their cytotoxicity, their ability to kill cancer cells. They essentially turned into superior tumor killers.
So you're reprogramming the immune cells by cutting off one of the cancer's fuel lines.
Kind of, yeah. And they showed this worked in vivo in actual cancer models using small molecule drugs to inhibit ANT2.
Incredible potential there. And speaking of advanced tools, we have to talk about AI, DOLPHIN.
Yes, DOLPHIN, DOLPHIN. This is an AI tool designed for analyzing single-cell data, but with a really fine-tooth comb.
How so? What makes it different?
It analyzes RNA sequencing data right down to the level of individual exons and splice junctions. That's much deeper, much more granular than most traditional methods.
Okay, so it sees more detail. What's the practical upshot of that?
Well, in their analysis, it identified over 800 potential cancer-associated genetic markers that older methods had completely missed.
800 missed markers. Wow.
Yeah. And the bigger picture here is that this kind of deep analysis lays the groundwork for creating virtual cells.
Virtual cells, like simulation.
Exactly, to model and predict how cells, including cancer cells, will respond to different drugs in silico, on a computer. This could massively speed up drug development and optimization, potentially skipping a lot of laborious, time-consuming lab work.
Just simulating drug effects before you even get to the lab bench. That's a paradigm shift.
Yeah.
Okay, one more preclinical piece. Durable gene silencing with Epigenetic.
Right, Epigenetic. These are engineered proteins based on TALE technology, TALE. What's remarkable is they can silence specific genes, like oncogenes driving cancer.
Okay.
But they do it durably without actually altering the underlying DNA sequence itself. It's epigenetic regulation.
So you're switching the gene off without cutting or changing the DNA code. That sounds safer.
Potentially, yes. It reduces the risk of genotoxicity, unwanted DNA damage that can sometimes come with gene editing tools. Plus, it might be reversible. And the delivery method is also important.
What are they using for delivery?
They're using LNPs, lipid nanoparticles. This avoids the complications sometimes seen with using viruses to deliver genetic material. So the LNP delivery makes it attractive not just for cancer, but maybe for cardiovascular diseases too.
Fascinating. Okay, so let's try and tie this all together. What's the big picture here?
Well, you're seeing this amazing convergence, aren't you? In the clinic, you have refined treatment selection with tools like PAM50 and more aggressive, effective combinations for advanced disease.
Right, optimizing what we can do now.
And then looking forward, you have these revolutionary tools emerging from the lab. Things like metabolic rewiring with ANT2 to boost immunity, AI like DOLPHIN to accelerate discovery and simulation.
And epigenetic editors like Epigenetic offering precise gene control without DNA damage. It really feels like progress on multiple fronts simultaneously.
It really does. Which leads to, I think, a really provocative thought for you, the listener, to consider. Given how fast these preclinical breakthroughs are moving, the AI, the metabolic targeting, the epigenetics, how quickly could these actually translate into the clinic?
Right.
How soon might they fundamentally change what we consider the standard of care, especially for high-risk prostate cancer? Could it be sooner than we think?
A powerful question indeed. Something to definitely keep an eye on.
Absolutely.
Stay strong and fight on.